Pride of Calgary, Alberta. Not just Tegan, not just Sarah.
Tegan and Sarah.
Awesome.
Welcome, everyone.
Good afternoon.
Hello. This looks beautiful.
I don't know if that is a muscle. I think that's Audrey, too.
Feed
me, Seymour.
Feed me, Seymour. No, Marwan's been busy, and it's fantastic. And I'm going to say that it is a freshwater mussel.
I think it's a carnivorous plant.
Do you think it's a carnivorous plant?
Uh-oh. There's the base.
There's the base. Uh-oh. I might be losing. No, but look, there's the fish.
When I was younger, just a bad little kid.
Okay. Hi, everyone. How are you?
Hello.
How are things? Are you okay? Jump
And
in
be
the chat.
honest, we know they're not good.
We know they're not amazing. So, like, new bar.
How are things considering?
Yeah, we're okay. Considering. Yeah, things are not easy, and we've got each other. We're in good company. Everything's fine. Everything's fine. I feel like that dog sitting in the fire.
Got you, babe.
Wait, where's my mug? I need a mug. Everything's fine.
Let me open my...
Yeah, there we go. Your ginger ale?
My ginger ale.
Oh, that's cool. Okay, super good. Some of you are still coming in, so we'll not say anything super important for another minute or so. I guess super important, but we have a whole slide for it, so we can maybe start talking about that. Is your topic test number 1 is coming up. And there'll be obviously all the information that you need about it. We haven't written it yet, but because everything's fine.
It's all right.
It's all right in here.
It's all right in there.
Yeah. Wouldn't it be great if we didn't? Okay. So.
If we didn't have anything in here.
Yeah.
We'd be administrative.
We'd be running this university. I'll just come out and say it. Okay. So yes. First of all, full on, we know that test taking can be anxiety inducing. And so all the things that we're doing are there set up to try to reduce anxiety. So yes, you're still coloring, but let's screenshot this and then maybe we'll go to the next slide where the details are. Okay. Because I think these screenshots are beautiful. So I'm screenshotting it, and now I'm going to clear it, okay, so that I can go to the next slide with the information about the topic test. Okay, clear all drawings.
Before you get on,
what?
2 things. One of them is as you can see at the bottom down here, we've gone back to the Google seems to have fixed it. If the transcription disappears, please let us know in the chat because we'll probably be staring at you.
Yeah.
And so we might not notice if it drops away. That was the problem that it was giving us before.
Yep.
And the other is that if you got the word, I'll put it in the chat.
Oh, good. Okay. Slide number 2. Not all the information, some information. So it's coming up. It's on a Friday. All of the topic tests are on Fridays. And you can log in to do it anytime between 9 and 6 p.m. Now, 6 p.m. is deliberate. It is not our usual 10 p.m. deadline. It is 4 hours earlier because we are standing by to answer your questions by email if you have any about the topic tests. So we are going to be available from 9 until 6. The early adopters, the ones that come in at 9, if there's like a little technological glitch or something, like let us know and we'll go in and fix it and we'll reset it the way, whatever. Just let us know if something doesn't work for you or if something seems strange, and we can do all sorts of stuff behind the scenes and after and whatever. So with 1,000 people that are going to be writing a test, we have a lot of eyes on the test, and so we have lots of opportunities to make corrections. So if you do need to get in touch, please go ahead, and that's why we offer it from 9 until 6. It's a 45-minute test that everyone is going to have 3 hours if they want them. We're not recommending that you spend 3 hours.
Not at all. Please don't misunderstand that.
But if it means reducing your anxiety because you're going to do it slower and not like speed read or you're going to reread things, then amazing, because we're not testing your ability to solve an evolutionary problem in 2 minutes. In fact, no evolutionary problem has been solved in 2 minutes. By definition, it's a slow process. So 3 hours. And it's open book, whatever book means to you, but no websites where academic misconduct is facilitated. We do monitor them, we do check, and so please just let's not. And then if everything goes well and we have no evidence of academic misconduct, we'll continue doing the topic tests in this way. If it turns into a disaster and there is evidence of misconduct, then we're going to have to figure something out. And I don't want to go there because I like these tests. These I'm kind of proud of. I feel like they really do evaluate your ability to apply things, and that's what we're going for. So if everyone's cool with it, then let's just make sure it goes really well, and then we can continue doing it in this way.
Some conversation about the topic test in the chat.
Okay.
Is it multiple choice? As we said, we haven't
We
written
don't know
it
yet.
But yeah, it'll have some multiple choice. It might have some true false. It might have some short answer. It's kind of in that realm. And then people had asked, is there a practice test? Is there a practice topic test that we can do?
So yes and no. We have better than that. So practice tests, actually, if you look in the literature, don't actually help students learn the material. They do reduce anxiety because you get used to the way that the prof writes the thing. So what we did was we took that idea of anxiety reduction, and we develop, as you can see there, the asynchronous study tool. So we will be posting that. That will be available on Monday. And the more that you actually use it, the better learning you will get. And so the idea, of course, is that you have to be active in your learning. This tool really encourages you to be active. And all of the instructions and everything will be there. It's this Google spreadsheet thing. It's really cool. It's a lot of fun, and we monitor it throughout the week. And yeah, so the more active you are, the better, but it requires like a bunch of you to be active and engaged in it. So it's kind of like this asynchronous space for you to study together. And the more you use it, the better it will be.
Yep, I think that's right. And so the idea that you may have had reinforced in high school and other classes that read your notes, reread your notes, reread your note. I'll even offer a counterpoint. Fine, do that. But that's the end of the beginning, not the beginning of the end of studying.
Yeah.
When you've read your notes again, when you've rewritten them and you distill them, that's a common starting point. What you don't ever want it to be is the end. Then when you've done that a couple of times, then you're ready to participate in the asynchronous study tool and ready to participate with the online or the actual community that you have in residence or in digital space.
Well said. Yes.
Sometimes it happens. It's like monkeys in a room with 1,000
typewriters.
typewriters.
Things.
To me, not to me.
Okay, super good. So yeah, and of course, if this course is not your priority, you'll do okay, even if you don't spend too much time studying. But I know that studying and getting good grades is a priority for the winter semester student cohort. So here are all the tools that you need to do well and to get through the applied part of this course.
Just a clarification question about sites to not use. Are we allowed to use CourseLink? Yeah.
Yes.
Yeah.
No, I'm talking about Chegg, but I don't want to give you like a full rundown of all of the places.
Might not have run them.
Don't go there. Plus, I was there recently, and some of the answers aren't right.
People that populate that are often not doing great.
Yeah, so don't go there. Don't do it. With 3 hours to be able to figure it all out, you should be good. I am in a bit of a conversation with them about copyright infringement and stuff like this, and so just stay away from there. All of the materials that you need, including definitions and stuff, sure, Google, like Wikipedia is your friend. Use it. But don't go and don't share answers or post answers or ask other people for answers.
So those are the places we're talking about. Not really. There's, can we expect to see questions about muscles on the test? We're not asking you to memorize things about muscles. If we give you questions about muscles in the question, will be all the things about muscles that you need to know. It won't be like a gotcha, like, did you remember how to spell Veliger? Did you? Because it's like, the internet will tell you. It'll say, if you spell V-I-L-I-G-E-R, it'll say, did you mean V-E-L-I-G-E-R? And it's like, I did. I did, in fact. So it's like, we're not
no trivia. No trivia. Anything that you can Google to get information on will not be tested. But if you want to Google stuff to get information so that you can build an answer to your question, that's cool. Okay. You'll see. And we have 3 of them.
Yeah.
And so the next time we do this, you'll be like, oh, yeah, we know what Jacobs and Smith are going to do.
Just the other reminder, or the other kind of reiteration that we've done in a bunch of cases, which is kind of in general, it's like what kinds of questions. The kinds of questions are going to be like the homework questions. Like those are really the kinds of questions.
Yeah. Okay.
So,
are we descended from monkeys?
Speaking of monkeys, in a room.
Okay. So, we're going to be focusing on tree thinking and tree reading and understanding trees. We kind of got you that little introduction in the pre-lecture stuff. So let's kind of practice this together and make sure that it's a little bit more clear.
Is this our last evolution?
No, we have one more evolution class. Yeah. Because it says here in the slide, the last class will be Monday.
I was looking deep into
my eyes.
Bumper stomper bumper.
Okay, cool. So, we are going to kind of really practice and get some feedback from you on how you're doing with the whole tree reading thing. It's kind of a skill. It's not super easy, and it is so cool. The amount of information that you can get from an evolutionary tree, and we want to kind of open your minds to all of the different ways of looking at these trees and interpreting.
Can I make a prediction?
Please.
We may have, because of being nearly 2 full years into the pandemic, a very more tree literate than average cohort.
You're so right.
Perhaps.
Okay. Let's see.
Yeah.
It turns out that the internet and the interwebs is not super evolutionarily literate when it comes to understanding what it is exactly. And so I always do Google image searches when I search the internet. And so this is what I get when I search evolution.
Now I see one thing in there that's pretty special.
This one?
Yeah. No, the beak of the finch.
The beak of the... Oh, we're going to talk about the beak of the finch.
And we'll get to that later on.
Okay.
But boy, oh boy, those lines are...
Yeah, so it's this linear idea, the sort of the idea that we are descended from monkeys in a very linear kind of sense is really so wrong. And we want to kind of explore that today, right? We want to kind of correct that image in our minds when we think about evolution. And we're going to do it with 5 lines. 5 different colors, each color representing a modern species. Okay, and what we're going to do is we're going to build a tree. Now, this is the point. This is a really critical point. Remember the last time when we were like, hold off on your stamps, just hover, and then stamp when we say 3. Can we do that again? Because that works super, super well, and it lets us know your level of understanding and how you're doing and all of that stuff. So we can clarify misconceptions if they happen. So get your stamps ready.
But don't stamp.
And if you really need to stamp in the margin.
Just no stamp.
Stamp in the margin.
Stamp off.
There we go. Okay, good. So what we're going to do is we're going to like put up a blank tree, and we're going to give you a statement, and then we're going to ask you to hover, and then on 3 stamp so that we can test some things, okay? Are you ready? Let's go ahead and clear, and then, okay, hold off, don't stamp. Okay, and what we're going to say is yellow is the outgroup for the other 4. So if you wouldn't mind picking maybe a yellow stamp, I think the star is yellow. So pick the star, and on 3 stamp for me what yellow would be if it's the outgroup. 1, 2, 3. Okay, super. That's really good. Okay, excellent. Yes, so most of you have it correct. The outgroup is the branch or the species at the end where the branch sort of branches off first from the base of the tree, right?
The earliest branching.
The earliest branching, that's right. Okay, that's great. Okay, so hold off. I'm going to clear.
We're going to clear several times.
Several times, probably. Yeah, it's okay. It's just a lot of you, so it's fine. Okay. So if we were going to start to like construct this tree, we've basically taken the yellow off at a very early stage from the base of the tree. Okay. But we haven't resolved anything about the other colors yet. So let's start to do that, right? So we have one part resolved, the outgroup. And now we're going to go to the second step. So again, like hold off on your stamps, okay? And I think you want to pick a heart for this because that'll give us the red color. Yeah. So if blue and red are sister taxa, which one of these is red? Which branch is red? Go ahead and do that now. Excellent. Okay, that's super helpful. So most of you are getting the correct answer, that short branch here at the end. Some of you are getting the one that's just next to it, and we're going to clarify that exactly. So don't worry, by the end of this, you're going to get this. A sister taxon is one that is most closely related. And so what you're looking for are very, very short branches, right? And the reason why it's this red one here that's getting covered with all of the right answers is because it shares the most recent common ancestor at this node with blue, okay? This one, this common ancestor, this branch, is an older common ancestor. It is not the most recent common ancestor. Super. Okay, are we ready to continue? Okay, so red and blue and sister taxa, we've resolved this relationship, but we still haven't resolved two of them. So let's go ahead and resolve the relationship or the order in which black and green are going to be branching, okay? So green is more closely related to red than it is to black, and I think green is the check mark. So go ahead and get your check mark and tell me where green is. Excellent. Okay. Super good. Okay. And then by default, the remaining gray one is going to be the black. Yeah. Good. Wonderful. Okay. So that's kind of basic, and you're doing very, very well. That's fantastic. So we want to kind of up our game a little bit.
It's not basic; it's fundamental.
Fundamental.
It is those were tricky things that you can find mistakes of in the literature.
Yeah. Particularly
the sister taxon.
Yeah. Okay. Super good. That's right. It was fundamental. It's kind of like tree reading. Basics. Yep. And then from there, once we understand that, we can then start to infer all sorts of sort of secondary relationships and things like this.
Those are the fundamental steps. Now let's dance.
Let's dance. Okay, good. Okay, so let's just resolve this tree though. Here we go. Just by showing what it would look like, we've colored it for you. Thank you for coloring it though. That's great. Okay, so green is more closely related to red than it is to black. Right? And what we're looking for, and this is a very popular misconception, it's not the number of nodes, okay, when we're talking about a tree that depicts species, it's where they are in the tree relative to the other branch, okay? So these two are very, very closely related. Here, wait, let me get onto my, here. These two are very closely related because they share a recent common ancestor. Red and green are more distantly related because they share a more distant common ancestor with respect to time. Okay? Cool. Some people just go, okay, well, black is only one node away from, don't do this. Black is only one node away from green, but black is like 2 nodes away from blue. So it's, anyway, don't do that for a bunch of reasons. But the reason why that misconception transfers sometimes is because if you're doing a phylogeny of mutations, like single event mutations, then you kind of can do that, but it's complex and just don't do it. Not yet. Okay. Where are we going? Now. Okay. So there are different ways of depicting a phylogeny. Sometimes it can be the like football goal way, and other times it can be this more triangular, kind of slanty way. They're the same information, the same branching. It just really depends on what you prefer. What do you prefer?
I prefer the, like if I was to draw something.
You draw these, right?
I would draw those.
Yeah, okay. I kind of like these, but just because it's more efficient design-wise. But Anyway, okay. The other thing that's really important to remember is that across here, across the top, the order in which things are placed contains no information, like no readily available information, okay? The information about the relationship between the species is contained within the ordering of the branching. And so what that means is you can take this tree that we showed you before and you can twist the nodes to change the order across the tips without changing any of the information within the tree. That's an important thing to keep in mind.
Nodes can rotate, just as branches move and twist in the wind on a phylogenetic tree.
Yeah.
They move as well. Rotations contain no information.
That's right.
So remember we've talked about when we were introducing kind of words into your Scrabble lexicon, into the bag of words in your brain that you're going to pull out. We talked about monophyletic, paraphyletic, and clade. So we're going to review briefly what those are, just in case that was the first time this kind of systematic terminology had come up. So the clade, this is this group of organisms, so maybe of species, but of taxa that it has to consist of the common ancestor and all of its descendants. That's a clade. Now used correctly, it's synonymous with monophyly. Monophyletic, so saying a monophyletic clade is actually redundant. Monophyly, clade, same thing. Okay?
Yeah.
And the clade has to include, I can't see, but two things. What two are they? The common ancestor and all the descendants.
So, and I think we need to clarify what we mean by all of the descendants. What we mean by all is 100% of the descendants. Okay? So you can't not have some of them represented and then it still be a clade. And we'll show you why that distinction matters.
So there's monophyletic means clade. Yep, clade means monophyletic. So there's para and polyphyletic. Those are ways that taxonomic kind of a grouping or a taxonomic hypothesis can be not monophyletic. For you, and I would say for most evolutionary biologists, the difference between the two of them is not super important. We just went over this in the second year invert course. I would suggest to you that what you need to be able to do is to identify where monophyly is broken.
Yeah.
How it's broken is going to depend. Sometimes it depends on phylogenetic hypotheses that were wrong because we didn't know things at a time. There's an example of that actually on the screen. You'll see it in a second. And sometimes it depends on us getting traits wrong. And then traits are kind of mapped onto a phylogeny incorrectly. The first one tends to be paraphyletic. The second one tends to be polyphyletic. So paraphyletic is an example where you're missing, at least most commonly, missing a descendant.
Right.
We have an example of that coming up.
To clarify, for the purposes of this course and our testing and all of that, paraphyletic, the definition of paraphyletic is basically not monophyletic.
Yeah.
Cool.
So this is from that source of biosystematic information that we talked about earlier that you can use on your topic test. This is from Wikipedia. So we have here shapes onto a phylogeny. Okay, so we've got one of them in yellow that includes all of the reptiles, all the crocodiles and the turtles and the birds. So that's a monophyletic group. Reptiles, just as we would use it historically, doesn't include birds. That's a paraphyletic grouping because it excludes one of the descendants from that common ancestor. And then we have a group or a trait in this case, which is polyphyletic, which is red, warm-blooded. So red, going from mammals and kind of selectively jumping across to birds. So those are the two examples that I was talking about a second ago. The word reptile we made up to describe how we understood phylogenetically, taxonomically, the world. We got more information. We now know that birds ought to be included in that. So that's a taxonomic word that with more information we now understand to be paraphyletic. Warm-bloodedness mapped onto this phylogeny we understand to be polyphyletic. So
there you go.
Ta-da!
Wonderful. Okay, so let's see if we can apply that to
So a
simple
C
problem.
Danny 07. Yes, reptiles not a clade.
Not a clade. Okay. Here we go. Here's a clades question.
Yeah.
Please don't annotate on the slide. Go ahead and just answer the question in the poll.
So basically, crocodiles can fly. Captain Hook would be so disappointed if that was the case. If I remember my Peter Pan correctly.
Super. Okay, just a few more seconds. Yeah. As you prepare for the topic test, play with this diagram. You can ask yourself all sorts of questions like, how many clades, by definition, are represented in this phylogeny? If you want to kind of extend your studying a little bit. Okay, say another 10 seconds for the question. Okay, wonderful. Good. Okay, super. 90%, 89%. Share results. Yellow and black is the correct answer. So very well done. That's super. So yellow and black as a grouping are not a clade because they are not monophyletic. They do have a common ancestor, which is here. But if this is the common ancestor of yellow and black, which it is, then a clade has to include the common ancestor and 100% of its descendants, which means that we miss out on green, red, and blue. So just yellow and black together are not a clade, okay? And all of the other ones are. Red and blue have a common ancestor that includes then all of the descendants, right? Does that make sense? Okay, cool. Excellent. There we go. Okay, super. So just to kind of clarify, at each of these nodes then, what we end up having, or the nodes are created sort of in real time when there's a speciation event, and in real long time, like a long time it happens, okay? But what is captured there in the information within the tree is a speciation event. It means then that, you know, we have 5 species here represented, 5 extant species that are living today. But within all of these sort of trails to get there are thousands and thousands and thousands of other species that have evolved and gone extinct. And at this point here was a speciation event that led to the outgroup branching off and going in its own kind of, in its own direction, right? Its own kind of way, its own history. But certainly captured within this line is the potential for other speciation events and other things. And we'll kind of bust open one of those lines for you in a little bit. The important thing to note, though, is the way that the speciation event occurs, and speciation event is the evolution of a new species, is going back to gene flow, and there's a barrier to gene flow. Okay, so just to review, what's gene flow?
So gene flow is when there's the passage of genes, of the sharing of alleles between populations.
Cool. There you go.
Maneuvering.
Immigration.
Mating.
And emigration that results in mating, right? The swapping of genes, the homogenization of the population, the two populations, right? So if there's a barrier to that, it means that they're evolving over here in a randomly dictated kind of way that's based on selection pressures that may be different from the population that's evolving over here, and eventually over time they get different. That's a speciation event. So there has to be a barrier to gene flow in order to be able to have the conditions that might, not necessarily, but might lead to a speciation event. So let's play around with that a little bit. More
Quizzing.
questions. Okay, tree reading. There you go. I'm working through it in my head.
I can hear it.
Oh, you can hear? Do you hear the creaking?
Wicked.
Let's see what they say. Let's see if they're right. Some students find it helpful. Not my brain. Okay, just about maybe 20 more seconds.
The rate at which you're voting suggests that we are kind of proceeding along. This is like, okay.
Yeah. It's good. You're doing great.
Papa,
Salut,
I
monsieur.
saw this on your.
Oh, merci.
Oh, merci, monsieur.
I saw it on your desk.
Okay.
Merci, monsieur.
Voilà. Okay, easy. Okay, so 85% of you. We'll give you just a few more seconds, see if we can get that number up a little bit. And
C'est quoi?
wonderful. Okay, that's great.
Mais peut-être c'est
So
en
I'm going to end it right now and share the results with you. Thank you so much. And the last one is the correct one. Let me just double check though. I'll walk through it out loud if you'd like. The tree on the right shows that frogs are more closely related to fish than to humans. Okay, so if I'm going to evaluate that, I'm going to look for where frogs is and where the common ancestor with humans is, because I'm doing humans, frogs, and then frogs, fish, right? So human is here. I go down and I look for where it intersects with frog, and that is right here. So that's my common ancestor that I'm going to be comparing it to in the comparison. And then I'm going to look for the common ancestor between the fish and the frog, and that is fish and the frog. Here is where they intersect. So I'm comparing the relationship between two things that intersect here versus here. And this one is more recent, which means that whatever comes from there, thanks Hannah, that's super helpful. Whatever comes from there is more closely related. So the human is more closely related to the frog than the fish is to the frog. The fish and the frog are more distantly related. Okay? So shows that they are more, so frogs are more closely related to fish. So that is a false statement, and we're looking for the false statement. So there you go. That's how you get to it. The rest are true. So work through them, figure out why and how you can make them false, how you can make the false statement true, in order to really kind of use this as a study tool to be able to help in your understanding of the concepts.
And really involve your community.
Yeah.
Make these questions for
Yeah.
yourselves.
Yeah. So that's what the asynchronous study tool is, basically. We're going to give you stuff and ask you to write true, false statements. And they can be true or false. The idea is not to tell us what it is, we are going to vote on whether we think it's true or false. So the more precise you can be in your language, what you want to do is not write something that is so obviously true or obviously false. So you'll be able to practice that. Anyway, it's a good tool. I like that tool. Okay, cool. Next, is this a real tree?
It is.
I love this tree. Why is this a real tree?
This is a real tree because it's a tree of deuterostome, so it's within the phylogeny, the tree of animal life, in which we are one small branch. We share that are part of the tree with all of these other animals. These are the closest invertebrates to which we are related: the crinoids, the sea stars, the sea cucumbers, and the urchins. These are all things. These are all amazing things that we ought to dedicate more time to. But the point on this slide is on the left-hand side of the slide, leading to that human baby. It is that there's a whole bunch that is shown. Just as in good art, there's a lot of art that's done in the space that's left behind. There's the active lines, but then there's the space. We tell a phylogeny is a story. A phylogeny is a hypothesis of relationship between taxa and species or other levels of taxa. But it implicit to that is that there are a lot of lines that are not there. And there's a lot of change that happens along that branch. So in this case, all of the change amongst the vertebrates that has led to primates, all of the change amongst the primates that has led to Homo, all the changes amongst Homo that has led to Homo sapiens is not there, not drawn out in this clade, but it is there within that branch. It's kind of fractals. There's yaks all the way down, like if you zoom and enhance and zoom and enhance,
yeah,
and it's there.
Good. The other thing that's important or sort of information that you can glean from these types of trees is that it moves along an axis of time, right? So if we have our phylogeny stacked up this way with the extant species along the top, then time is here along this y-axis, right? It means, though, that we need to say things like humans have been evolving for the same amount of time as fish, right? Because they are all extant species, all descendant from a common ancestor. Now, their rates of mutation may be different, their rates of speciation may be different, but certainly all of them have been engaged in the same time of evolution.
Can I say something about, you had asked earlier about different representations of trees. This is one of the reasons that I like this representation, because A is equal to G plus F plus D plus D.
That makes sense.
It helps people understand that when we're talking about extant species at tips, this is the same amount of time.
Okay. I'm becoming team football goal now.
It's also easier for me to draw.
Super. Okay. So, are we descended from monkeys, right? This was our original question. And the answer is, it depends. No. Yes. It depends. What does it depend on?
So not any of the modern monkeys that we see today, because
right.
They're all here with us today, right?
Yes.
We've all been here for the G plus E plus F plus D amount of time.
That's right. But we're all descended from the same common ancestor, right? And so if you consider this thing down here to have been a monkey, then sure, we're all descended from monkeys.
Yep.
If you consider it to be the common ancestor of modern day monkeys and apes, then the answer is no. So why are there still monkeys? Because they're still evolving. But we have to be able to switch our sort of linear understanding of evolution to something a little bit more nuanced and way cooler.
It's certainly not progressive.
That's right.
And so one of the last stories that we have prepared today is an example that largely predates COVID, where a lot of tree-based thinking is used kind of daily now, nightly on newscasts. I've even seen it on late night TV, on things like The Daily Show and The Colbert Report and that kind of thing. So the power of phylogeny as it affects human health was one of the first places it was shown here was in this kind of tragic story. And when you get the slides later on, you can click on any one of these pictures and go to links of the original papers and some discussion about them. The story was essentially in the late 80s and early 90s in Florida, when HIV transmission was
Poorly understood?
Super poorly understood, but even vilified. And of course, that clearly not helping our understanding of it. There was a case in Florida where 7 people reported caught HIV, developed the symptoms leading to AIDS, and some of them died. And there was a whole outbreak of people. There was clearly an outbreak, and the community, the Floridian community, was really confused as to how this could possibly have happened. And scientists from universities and from the Centers for Disease Control sequenced, DNA sequence, which was new at the time, one of the envelope proteins of the virus that seemed to be associated with AIDS and with HIV transmission. And what they found, they made these small, very simple phylogenies. And this is one, the bottom there is the phylogeny that was associated with the paper in Science a year later. And I'm going to break it up for you a little bit. I'm going to do exactly what I said I could do. I'm going to click on the wrong part of the screen and demonstrate for you that you can go to the paper by clicking on it. I'm going to go back here and click somewhere else, less sensitive. So you can actually, one of the neat things about DNA-based information leading to trees is that it should be transparent and reproducible. And what I'm doing here is kind of showing you that. I'm demonstrating that because later on, if you click on the text at the top of that, you can go and look at the DNA sequences from this paper. I did. I'm showing you them on the right here in a picture. So this is the DNA sequences of that HIV virus from top to bottom across Florida, all of the known cases from Florida, including the 7 who were confused as to why the mystery 7 as to why they had it. And the question was, where could they have captured it from? Where could they have been infected it from? Now, some of the detective work at the time suggested or turned up that they all shared a dentist. And the dentist, in fact, himself died from AIDS, developed HIV and died from AIDS. And so the CDC scientists sequenced the dentist, sequenced all of the patients, and included sequences from the rest of the known diversity of HIV lineages. That's the tree from top to bottom, to try and answer the question, to use phylogeny to answer the question, did the patients, were they infected in a clinical setting by the dentist? And so I'm going to ask you to use your annotate tools here in a second and say, so if the HIV infection that the 7 patients, if it came from the dentist, where on that phylogeny, show me some circles or some stars, where do you think, if this is the dentist in the red star, where would those patients have been found? Screen grab that. That's awesome.
You want to?
Yes, please. You're totally right. So out there in Zoom land, if you're in the same room with one of your peers, if you could please high five, like loud enough so that we can hear. That's right. That's exactly right. So and this was used in and tested, and this was the first demonstration of the kind of the power of phylogeny, the power of molecular phylogenetics as it pertains to human health. Now, of course, in the last 2 years associated with the COVID-19 pandemic, we've seen this kind of science deployed all the time when we think about, well, what was alpha like compared to delta? What was delta like compared to Omicron? All of that.
That's right. And so if we take a look at the paper, at the data here, what you can see then is that the dentist, right, becomes part of the sort of outgroup, the first one of the clade that are then the rest of the patients, right?
So this is a tragic story, and those patients, the terminology there inside of the box that Dr. Jacob just formed, they have released them and their families because some of them have passed on from this infection. Some of them are still alive and are known and advocate.
AIDS activists.
They're AIDS activists now, and they lived through this. The dentist, as I said, did not. And this is one of the unknown or the mysterious parts of it. And the tragic parts of it is that, and there have been books written about this, is about how it happened, why it happened, As best as we can tell, the likelihood is that he did not know that he was infected. And one of the consequences of secondary infections associated with HIV/AIDS is the loss of sensitivity in your digits. And so the thought is that there was an error, a mistake, and some clumsiness that caused blood, and he didn't notice it.
Yeah, terrible story.
And boy, would it have been easier to do if it wasn't so vilified, if it wasn't so unfortunately politicized. And gosh, you'd think we'd never do that again, would you?
Huh, I wonder.
Yeah.
Okay.
I like that somebody just high-fived their mom.
Yeah, well done.
And high-fived the whole chat.
Yeah, okay. So the green box that I put around the dotted box here is now available to you in blown up form in purple, just to show you the clade of the patients, okay? Awesome. How do we apply this back to our muscles? Here are more things that you can do. Notice that we're not saying reread your notes. We're like giving you questions and activities. So here's an activity. Imagine that you have 5 species of muscles, right? These different colors. Pick whatever color you want. Pick a different species if you're tired of thinking about muscles. But have 5 different species of something. Put them all in a nice little row. And what we'd love for you to do is to go through this activity. So make at least 2 trees that are different, actually different, that show different relationships between or among species, okay, that show you different clades and all of that stuff. And then kind of walk through the implications of that, walk through the consequences, figure out who's more closely related to who, and that kind of deal. So basically like we had with the baby and the frog and the fish trees, but those 2 trees are actually identical. Make 2 different ones and come up with some different conclusions. Okay. And then here's another sort of series of 5 true or false statements. We want you to think about these things like in terms of true or false and applied. So that's like monster Easter egg, probably for the way that we write questions for the topic test. So here's some more for you. This one's a little challenging, so kind of walk through it. And again, there's no emergency in evolution that requires you to do this in 30 seconds. Take your time and work through it.
Only in science fiction.
Only in science fiction. We'll take this question up at the beginning of next class. Okay?
So, terminology question in the chat. Is there a difference between saying between species and among species?
Yes, there is a difference between and among, and it has nothing to do with biology and only to do with English usage and abusage. So between 2 things, among multiple things, more than 2. So if I have a conversation between Dr. Smith and me, that's between 2 things. If I have a conversation among my family, that means that there are more than 2 present.
So you'll often find them used synonymously. The counterpoint to that is within.
Ah, good.
So within species variance versus between species or among species variance. Those are the things that it's splitting up.
Yeah, that's right. The opposite of between and among is within. Yeah. Good. Right? Precise language. And if you ever get confused about this, Google the definition, right? It's really important, and it is important, right?
Did you notice my carriage return? Like I'm very
That's
old.
so old school. So a typewriter was a thing that we used to use. Okay, good. Bye, everyone. Thank you so much. It was super fun.
Be kind to yourself. Be
Yeah.
kind to other people.
Okay.
And
We'll be here
see
for an
you
hour.
here. Yeah, we're going to turn off and then turn on again.
If you need us, we're here. Take care.
Got all involved in not thinking about what was coming, and then I was like, oh yeah, the HIV story. Where does it start?
Yeah.
Oh, sorry.